The Pathway Tools Omics dashboard is a new tool for visualizing omics data, such as gene expression data or metabolomics data. The dashboard facilitates a rapid user survey of how all cellular systems are responding to a given stimulus. It enables the user to quickly find and understand the responsive genes within one or more specific systems of interest and to gauge the relative activity levels of different cellular systems. The dashboard also enables a user to compare the expression levels of a cellular system with those of its known regulators. The data is presented as a set of top-level panels covering different areas of cellular function, such as biosynthesis, degradation, energy metabolism, and various non-metabolic functions. Each panel is divided into several component subsystems. For example, here in the biosynthesis panel, we show amino acid biosynthesis, nucleotide biosynthesis, fatty acid and lipid biosynthesis, etc. In this example, we are looking at 5 time points from an Escherichia coli gene expression time series dataset. The values are all log ratios of fold change relative to control. Positive values mean that a gene is overexpressed relative to control, and negative values mean that a gene is underexpressed. This line summarizes the data for a single time point for all the genes involved in amino acid biosynthesis. The large dot is the average value over all those genes. Each small dot represents a single gene, and the line connecting them shows the range of expression values over all the genes in this system. If we click on this plot, we can drill down to show a detail panel for amino acid biosynthesis, broken down into plots for each individual amino acid. Drilling down even further produces a panel that shows the expression values for all the genes in the arginine biosynthesis pathways. Now that you have a general idea of what the dashboard is all about, I'm going to show you how to upload your data set to the dashboard and then introduce you to some additional features and options. To invoke the dashboard from any biopsych web page, use the command Omics Dashboard in the Analysis menu. There are 3 options. If you have recently, in the same session, uploaded a dataset to another of our omics viewers, such as the cellular overview diagram or a pathway page, you can opt to reuse it. If you have a recently visited smart table that contains expression data, you can upload from there. However, today we are going to upload a dataset directly from a file stored on my computer. This is a tab-delimited file with gene names in the first column and RNA-seq time course data in the other columns. The format of the file and the fields that need to be filled in are the same as for all our other omics viewers and are described in another webinar, so I will not go into detail here, other than to mention that the data values are counts, not ratios, so these are absolute values. Once I submit the data, it can take a minute or so to generate the dashboard display. Please be patient. This particular dataset is a time series showing the changes an E. coli cell undergoes when transitioning from anaerobic to aerobic respiration, so we expect the most significant changes to be in the area of energy metabolism, but we might see changes in other areas too. Sure enough, glycolysis and fermentation seem to be decreasing, and the TCA cycle and aerobic respiration seem to be increasing. And there might be something going on with some of these other subsystems too. One disadvantage of showing all the subsystems within a panel on the same scale is that the graph can be distorted by a few high-value genes. This makes it very difficult to see if there is something interesting going on with some of these other subsystems. We offer a few different solutions to this. First, you can just magnify the panel by clicking on the zoom icon here. This helps a little bit. We can now see amine degradation and secondary metabolite degradation a little bit better, but some of the others are still hard to distinguish. Switching to a log scale by checking the box here solves the problem. You can now see the trend in fatty acid degradation, as well as everything else. A third alternative is to set the scale manually using the Change Graph Scale command in the Options menu. Here, you can use the slider to change the maximum value on the scale. And click Set to see the results. This dialog also gives you fine-grained control over the size of the panel. Note that you can also bring up this dialog by clicking on any of the y-axis values. Clicking Reset reverts to the original display. Because different subsystems involve very different numbers of genes, it can be useful to show not just the averages, but also the sums of expression values for all the genes in a subsystem. In this mode, the averages and the individual gene values are still there, but now the display is dominated by these colored bars, indicating the sums. For RNA-seq data, this provides a measure of the total relative amount of resources the cell devotes to each function, and sometimes allows us to see trends that aren't particularly visible in the default display. For example, let's look at transporters. While the default display shows some movement, the trends, particularly for amino acid and secondary metabolite transporters, become much clearer when we show sums. This display also shows us that the cell is currently investing considerably more resources into transporting amino acids than carbohydrates or ions, which was not obvious from the default display. If we drill down to the gene level, we see expression data for each individual amino acid transporter. Because there are so many of them, and by default they are shown in genome map position order, it can again be hard to tell what is going on. However, by clicking on the sort icon, we can see a variety of sort options. If we sort these genes by decreasing change, that is, the difference between the maximum and the minimum time point values, we can see significant increases in several methionine transporters, as well as arginine, proline, histidine, and a few others. Note that when a transporter transports multiple substrates, you may need to mouse over the plot to see the full list of transporters. The log option, the show sums option, and the sort option can be set for each panel using the individual panel controls, but you can also change the default settings for all panels by setting them in the Display Preferences panel at the right of the screen. While we're looking at these controls here on the right, let's look at the series selection panel. This allows you to show only a subset of your time points or experimental conditions at a time. For example, we can compare just the first and last time points by suppressing all the intermediate ones. We can also edit the labels used in the legend here. And you can see the updated labels in the legend. We'll leave these other options for later, but for now, let's zoom down to a gene level panel. Here, we can see the expression values for all the genes in the TCA cycle. We can click on any gene to show the regular gene page for that gene in a new browser tab. We can also show the pathway diagram with the expression data, so you can see the genes and the expression values in context by clicking on the Show Pathways button. It may take a few seconds to generate. You can use the command in the Options menu to export this pathway to a pathway collage to generate a zoomable, user-customizable, and editable diagram. There's a separate webinar that describes pathway collages, so I won't go into further detail here. The expression value color scheme used for this diagram was specified when you first uploaded the dataset. If that range is not a good fit for this pathway diagram, as is the case here, you can change it with the Update Color Scheme command and select a new maximum cutoff value that will apply only to this diagram. You can also show operon diagrams for all of these genes to see how they're regulated. If you wish to see how the expression of transcription factors known to control these genes correlates with that of the genes themselves, use the Show Filter Regulators command in the Options menu. This operation brings up two insets. The first shows expression data for all the regulators of TCA cycle genes. The second allows us to zero in only on those genes regulated by a selected set of regulators. For example, FUR seems to be undergoing a bit of change. If I select it here, I now see only those genes that are regulated by FUR and their regulators. However, it looks like these genes are also regulated by quite a few other transcription factors. If I click IHF as well, I now see only genes that are regulated by both FUR and IHF. The remaining genes look like they're probably all part of the same operon. If I wanted to, I could bring up the operon diagram again to be sure. If I close this inset, all hidden genes are restored. Another useful operation in the Options menu is the Show Data as Table command. This generates a table containing the raw data just for the genes in this panel. The download icon lets you download this data to a file on your computer. This has been an introduction to the Pathway Tools Omics dashboard. Part 2 will show how we handle more complicated data sets, such as those that include data from multiple replicates or significance values for enrichment analysis, as well as how you can customize the dashboard. Before I go, I want to draw attention to this help link, which opens a comprehensive help document describing the dashboard functionality. This concludes Part 1 of the dashboard webinar.